Well, again, I want to extend my thanks so much, and it's so great to be here. And so I am not going to be really talking about mice right now. I'm going to be talking about humans. And I'm not going to be really talking about a lot of familial clustering or cancer predisposition syndrome. I'm going to be trying to address, using the genomic revolution and the data generated from that, what does P53 mean to you and me before we get cancer? And so this is my provocative question of, does the broader human population actually have innate or inborn differences from your mother, from your parents, in the P53-mediated tumor suppression? And I want to say, because obviously I'm here, I think we do, and I think it's becoming clearer. But the big question is, what can we do with that information, other than just an academic exercise? And so those are the things that I really want to end with. So I see this work, the laboratory that I run, is really contributing to a big field of how to tackle cancer heterogeneity. And I think we all know this from our personal experiences. Some people can drink and smoke all the time and never get cancer, and other people do. Other people are diagnosed with seemingly the same cancer, but some progress incredibly rapidly, and some don't respond to therapies, while other ones are cured, really cured from metastatic disease. And so there's a lot of different approaches to tackle this in the field right now. We're trying to take genetic information and really the inherited genetic information and try to use that to personalize therapy, because I was a Star Trek geek as a kid. And the reason why is that it's incredibly accessible and easily measurable. So theoretically, you can get information from any cell of your body and potentially use that, if indeed that information or that knowledge is stored in it, to help personalize therapeutics. Again, you can get it from any cell. Nowadays, with the new genomic technologies, it's so easy to measure it. And indeed, what's really the critical factor is the statistics to process it. And luckily, a lot of great quantitative biologists are coming into more mainstream applicable techniques. Moreover, it's ever present. So it's there before you get the disease, it's there when you get the disease, and when you're progressing and being treated. So if there is information to be had, we can have it before things get messy. And so our inspiration is what's happening in high-risk clinics today, which is actually pretty incredible. Everyone's heard, I don't think everyone's in the field, but Angelina Jolie and what she's done. And really that comes from this work. BRCA1 and BRCA2. We know that individuals who have certain mutations or families have an incredible high risk for developing breast cancer and ovarian cancer. Now those women, before they're symptomatic, can go into a high-risk clinic and they get treatment options. And these options can dramatically affect their overall survival, lifetime risk as well as survival from treatment. For example, Tim Rebick was one of the first people to show that prophylactic oophorectomies, so getting your ovaries removed, there's 80% less death from ovarian cancer, but also 50% less death from breast cancer. So just knowing that you have that inherited genetic mutation can drastically affect your lifespan. So we're not quite there yet in the broader population, but it definitely serves as a template and inspiration. So Arnie really, and everyone really, introduced this very well, is that P53 pathway is definitely a pan-tumor suppressor pathway. It affects so many different, it's really our innate protection against developing cancers in a lot of different types of tissues. And obviously the most dramatic one is that 50% of all tumors mutated and inactivated. And this is just to give you a little extra information. A lot of those mutations affect the way P53 binds to DNA, and that'll become important later. But what I do want to highlight, which hasn't been so highlighted, is that nowadays we can sequence genomes so easily. And like Arnie had introduced, we can get all these tumors, we can do full exome, full gene sequencing, and really look agnostically, so without preconceptions about what type of causative mutations we see. And what's clear is that the biggest and the largest mutated subnetwork circles around P53 in mutations. So it's not only the P53 gene itself, it's its large pathway or interaction network that also gets mutated in the broadest spectrum of cancers. So we're not just talking about P53, we're talking about its regulators and its downstream effectors. And they are hijacked and mutated by tumors and not just the P53 gene itself. And I don't need to introduce this anymore because Arnie has. We already know that the inherited genetics, again, in these about 500 families worldwide, that have this horrible cancer predisposition syndrome. And 50% of those are due to these damaging P53 mutations in their genes. So we know it's possible. So how about in you and me, where there's no familial clustering of cancer? What does the genetics of the P53 pathway look like? Because in fact, we don't know much about it. And I think that was due to a lot of different types of technologies that needed to be developed. But you've heard all day about these tumor mutations and this Li-Fraumeni syndrome and this beautiful, elegant work done in model systems that truly show these genetics are important. But what's really interesting is the clinical utility of this information is really lacking. And it's not just because of drug targets, it's also as biomarkers. We know it's always mutated, we know it causes cancer, we know it affects progression. But if you ask a clinician, when do you use this information, you don't hear much. And so a lot of clever people are trying to figure out what's missing, what do we need? And so what we're trying to contribute to is we really know nothing about the genetics of the broader population. We're not integrating that information at all. Is there a signal, and can it be useful? And that's really, in our P53 work, what we're trying to do. So a little background. What type of genetic variation do we have? If we take the genomes between two random individuals here today, what type of variation do we see? Now, one of the most frequent class, and that's a class we study because it's the easiest, is a single nucleotide polymorphism, or what you call SNP. And that's where you take one base and exchange it for another in the exact same location. And of course, there's millions of these in the genome. Something like 99.9% of all our genomes are the same. But if you break it down into base pairs, you still have way over 11 million SNPs, even within the same race. So this is a real challenge for the field of trying to find the signal from all that noise. Now, what changes this from just a mutation to polymorphism is that they're found in high frequencies. So if a mutation arises and then can get up to at least 1% in the genome, that's called a polymorphism. But I want to say in our work, we're looking at very high frequency events. So almost every polymorphism that I'm going to talk about today is at least at 10%, even up to 50%, 80% in populations. So it's really high frequency stuff. And now just in terms of jargon definition, SNPs travel together. They arise on a genetic background and they get inherited together. And those clusters of SNPs we call haplotypes. And sometimes I'll switch in between, so I want to make sure I've defined it. So there is precedent that this pathway can be very sensitive to these high-frequency genetic variants that we all have, or a lot of us have. And this is work that I first did when I was a postdoc in Arnie's lab over a decade ago, where we were able to show, everyone's heard about MDM2 now today, we looked in a very candidate gene way at the enhancer element in the MDM2 oncogene, and we found a change in its transcriptional enhancer that recruits SP1, a transcription factor, and upregulates MDM2 and leads to inhibition of P53 and an inhibition of the P53 stress response, which ultimately affects tumor formation. And obviously this was published decades ago, so I'm not going to go into the data, or a decade ago. But what's really been clear is that now, since that initial publication, we and others have shown that it really does have these P53 phenotypes of a pan-cancer tumor stress response. First of all, we've been able to show that it associates with an earlier onset and increased risk for cancers, but also different survival rates. And this is really in line with the fact that the stress response also affects things that affect tumor progression and response to therapy, so very P53. Moreover, the effects that we and others have seen have been in a wide variety of tissues, so really a pan-cancer effect. So this, I think, serves as nice proof of principle that this pathway can be sensitive to these genetic changes. But what really nailed it was a collaboration with Gigi Lozano, and really led by Gigi Lozano and Sean Post in her lab that we published about 5 years ago, where we actually were able to show that it's pathogenic in a mouse model. So she and Sean were able to make mice that were genetically engineered to either have the G or the T allele and follow them and look at sporadic cancers and also measure their effects in tissues. And so what was nice, they were able to show that indeed many tissues in the homozygous G allele mice did express more MDM2, less P53 levels, and a marked attenuation of the P53 response. But what was really important was these G allele mice got cancer faster. And this is just some of the survival curves that were introduced to you by Laura. The GG mice again develop and succumb to their tumors earlier than TT mice. So not only does it have P53 phenotypes in humans, we can really show that it's pathogenic in a mouse model system. And I think this still remains one of the only regulatory SNPs to be shown to be pathogenic in a mouse model. So P53 seems ripe for picking, for being sensitive for these SNPs and actually having a pathogenic effect. So now we come to work that we've done since the genomic revolution, where we can really have high-end sequencing at our fingertips. So the question that we want to address is, we know MDM2 can work. How many other P53 pathway genes have SNPs that actually affect cancer? Because we know somatically so many of the genes of the pathway are hijacked and mutated. And so to do this, what really opened the door for us is the Genome-Wide Association Study, or the GWAS. This is a case-control study design where you literally take hundreds to thousands. I think for breast cancer, we're already up to 40,000 patients. So really, really large populations. And you have controls who don't have the cancer. So you isolate their DNAs and you compare the sequences through genotyping, sometimes through more whole genome sequencing. And then you do a rigorous statistical analysis to make sure that you identify those variants that are either enriched, overrepresented, or underrepresented in your patient. So you're tying that genetic change to that disease, to that cohort. Now, this has increased. I think the first one was performed in 2005. You see this this is a number of SNPs that have been identified in GWAS to date. And these are all the traits that have been looked at, and these are the SNPs that have been identified in cancer. And you can see there's this exponential rise in the past 3 years. And this has really been due to, of course, larger cohorts and better genotyping, but also improved statistical methods. So we really have thousands of SNPs that have robustly associated with these diseases and traits. So we can start asking these in a genome-wide way, what's the influence of P53 and its genes on cancer and indeed in other diseases in humans and in the broader population? So we started curating in order to do these analyses. And of course, you have to stick in the same race, and because of a lot of historical reasons, Europeans are studied more frequently. And so if we just cut the GWAS data into European populations, we have 145 studies that have been done for cancer susceptibility, so just cancer risk. And the field has identified over 895 SNPs to associate with differential risk. And this can fall into minimally, it depends how you cut the deck, 17 different cancers. So we have blood cancers represented, solid tumors, and this is how we had a clinician help sort the cancers in different types that make sense clinically. And obviously, this is a number of SNPs that have been identified to really associate with differential risk. And of course, you can see there's a bias for the more common cancers, breast cancer, colon cancer. But we have a really nice spread, too. So we have a lot of blood cancers represented and also some more rare cancers. So, of course, we want to ask, do we see, if we think P53 pathway is so important, someone should be identifying P53 pathway SNPs here. And so the first step that we took, and this has been published, so a couple years ago, so I'm just going to breeze through it. But the first one we took was we wanted to see, is there a molecular signature that we can really tie to P53 pathway? And like we've said, the way it binds to DNA is crucial. The P53 gene is mutated in its DNA binding domain. So most SNPs are not in coding regions, they're in regulatory regions. So we turned it on its head and said, well, maybe response elements, their consensus binding site are going to be polymorphic. And luckily, there's this wonderful genomic technology called ChIP-seq, where we can get in vivo genome-wide maps of where P53 is binding. We can use position weight matrix to analyze its sequence and ask of all these cancer GWAS SNPs, and so this is called a modified circus plot. So you have the chromosomes head to tail, you have here all the cancer GWAS SNPs, all the linked SNPs, so the haplotypes included, and we took 11 different P53 ChIP-seqs we cast a really broad net. So we had ChIP-seqs from 6 different cell lines, 4 different tissue types, 7 different P53 activating treatments, found over 17,000 very, very significant peaks around the genome. And then when we overlay our sequence analysis, and I think anyone in the P53 field recognizes this, the tetramer binds to this region, and the big bases are the ones that make direct contact to P53. And so we asked, are there any of these cancer GWAS SNPs that hit these key bases in the consensus site? Now, very surprisingly, we only found one, but it was a very interesting one. So it's actually one of the most significant, strongest cancer associations that have been identified to date. It's been identified in 4 very large independent GWAS studies for testicular cancer risk, and it has one of the highest reproducible odds ratio in all cancer GWASs. Most SNPs only affect risk by 20%, but this one, males homozygous for this, have anywhere from 6 to 8-fold increase for testicular cancer. And this is just a plot showing that when you plot the odds ratio of all the loci found in the major cancers, testicular cancer is by far the highest. And this specific SNP is the highest. So there's only one polymorphic occupied response element, but it's a very important one. And of course, we did a lot of experimental validation, and the model ultimately is, as you go from the A to the G allele in Caucasian males, you bring down P53, upregulate more KIT ligand, which really does have a real experimentally tractable system. So we did this both in cell culture and in mice. So this is great, we found one, but really somatic genetics says we should find a lot more. And so we took a different approach, and we wanted not just to look at downstream target genes, so maybe the magic isn't there, but in the regulators or other components of the pathway. And so what we wanted to ask is, are cancer susceptibility loci, or these cancer GWAS SNPs, enriched in P53 pathway genes regardless of their molecular signature? Okay, now to do this, obviously we start with our cancer SNPs and we get their tags. Like I said, they don't travel alone. So we used the 1,000 Genomes Project Phase 1. We looked for very tight linkage, anyone who does this type of work. And so you have your cancer tag, and usually most tags in Europeans are minimally associated with 2 and go up to 10. So 1 becomes anywhere from 2 to 10. So we go from 850 to about 2,000, almost 3,000 SNPs. But now we want to attribute these to genes, and we're going to do this numerous ways throughout the talk, but the first one we did was just genic location. So we basically asked, do any of these SNPs reside in and around genes? And we look at all annotated genes in the genome, so they're about 23,000. And if they do fall in within a 10 kb region of an annotated gene pathway, we call them cancer susceptibility genes. So they're basically genes that have cancer GWAS SNPs. So very, very simple. And we go from about 23,000 genes to 460 genes. So about 460 genes have SNPs which have been identified robustly in cancer GWAS studies. Now, how do we go from genes to pathways? Now, what we do is we use pathway annotation databases because we want to compare one pathway to the next. So it has to be systematic. We can't just pick our favorite P53 pathway genes. We need someone who does this for a living to give us defined systematic pathway annotation. So I think most people who know this, almost everyone uses KEGG. This is a Japanese-based pathway annotation database that most people find the best. And they've annotated with confidence 214 signaling pathways in the genome. And these fall into almost all type of biological processes. The way they break it down is metabolism, genetic information processing, environmental information processing, cellular processes, and organismal systems. So we didn't cherry pick. We're looking at all well-annotated pathways. And our simple question is does the P53 pathway have more CSGs than expected by chance? So we use very simple statistics. So we use a hypergeometric enrichment test, which is basically a Fisher exact test. It's just computationally easier. And then, of course, we adjust p-values. We want to correct for multiple hypothesis testing, which is always a problem. And we do this using permutations, which is pretty much the gold standard nowadays with our computers. So this is what the P53 pathway looks like in KEGG. You have the stress inputs here, you have the key regulators here, and you have the downstream target genes. Actually, I think Arnie helped design this pathway. I think I remember them coming into your office. And then once these downstream effectors are activated, you have these cellular responses. So there's basically 67 autosomal P53 pathway genes. We're looking just at the autosomal genes because of a technical issue in the way GWASs are done. So out of these 67, we have 10 CSGs. So about 15% of the P53 pathway genes have SNPs that have been found in cancer GWASs. And what's quite interesting is that you have a lot of the regulatory genes that have been, and actually there is a significant enrichment of those being in the more regulatory mode, and then a couple key effectors. So is that a lot? Is that 14.9% a lot? So the first way we started to approach that is look at the remaining 22,000 genes of the genome and ask what percentage of cancer susceptibility genes do we have there. And you can see there's a 7-fold enrichment in the 67 P53 pathway genes. And this survives any type of statistical analysis. So now what about the other pathways in the genome? How about the other 214? And here we've plotted the adjusted p-value over fold enrichment, log scale, and the P53 pathways in red. So it's clear that, first of all, there's only 4 pathways that survive multiple hypothesis correction. So this is our cutoff for statistical power. But P53 is clearly the number one. But the other ones are actually quite interesting. We have adherence junction, PI3 kinase, AKT, and Wnt signaling. All pathways that have been shown in many different model systems to be important. And another P53 pan-cancer phenotype is a spectrum of cancer GWAS studies in which P53 genes were found were quite broad. So we have 8 different cancers in the way we've classified them. And indeed, this falls, here we plotted the number of cancers and the number of pathways that have been identified, and the P53 pathways in the top 2%, and only one pathway associates with 9 cancers, and that's the PI3 kinase AKT. So not only is it enriched, but it associates with a broad spectrum of cancers. Again, everything that we would have hypothesized. Now, another thing that, as many people have said in their introduction, is that P53 has been attributed quite beautifully to the pathogenesis of many other diseases. And these diseases include neurological, cardiovascular, infectious diseases, and all of these diseases have GWAS studies also attributed to them. And indeed, often they're easier to perform because it's easier to phenotype the disease. And so we took advantage of this and asked, does the enrichment we see, is that only in cancer? Or can we actually see it in other diseases? And so we were able to break it down into autoimmune and infectious, behavioral and psychiatric, cardiac and vascular, metabolic and neurodegenerative. What's really important is we get a significant enrichment in each disease. So there are significant, more than 2 in each disease, more than 2 pathways that have an enrichment in GWAS studies. But P53 is only significant in cancer. And indeed, we have absolutely no enrichment in the other diseases. So this would suggest that in terms of susceptibility, again, we're just looking at susceptibility, P53's main focus, at least to date, the way the GWAS catalog stands, is in cancer. So, of course, anyone who's a pathway biologist hates pathway annotations. Your favorite gene's not there. You think, you should delete one of the genes. So we wanted to make sure that the enrichment that we're seeing is not due to KEGG annotation. So there's basically two other pathway annotation databases that a lot of people respect and admire, and so we performed our analyses in those as well. The first one was BioCarta. And again, you see the same phenomenal signal in cancer and nothing in the other diseases. Again, P53 shining. And then the other one we used was Panther pathway annotation, and again, we see the same signal. And indeed, these are just subpopulations, the way Panther works, of P53. So only P53 is hitting here, and again, in the other diseases, we see no signal from P53. So everything that we've been doing so far has been using all SNPs. Now, we know, anyone in the human genetics fields know that the vast majority of SNPs do absolutely nothing. And indeed, many truly functional SNPs don't actually affect the gene in which they reside. You have distal enhancers, a lot of things can happen. So we wanted to make sure that we could see similar enrichments in those SNPs that have a molecular phenotype attributed to the gene in which they reside, their proximal gene. And to do that, we took advantage of some other genomic databases of expression quantitative trait loci. And this is basically where you do a GWAS, but instead of your trait being cancer, you do gene expression. And so what you do is you take, again, we're talking about hundreds to thousands of individuals. You take some cells or tissue, isolate their DNA as well as their RNA. You genotype and then do gene array analysis. And then you do the same type of GWAS statistics to identify those SNPs that reside in and around genes that associate with differential transcript levels. And so again, we cast a wide net. So we identified over 2 million eQTLs that were tested on 8 different cell types and tissues. So we used data derived from LCLs, PBCs, liver, lung, skin, B-cell, monocyte, and fat tissue. And again, what I want to highlight is that these are all normal tissues or cell lines. So we didn't want the complexity of the cancer genome. So these are all normal tissues. And what, of course, we wanted to focus in on are those genes that have SNPs that are eQTLs, so that associate with differential transcript levels of their genes, but also have been identified with SNPs and GWAS. And so what we're calling those are now eCSGs. So they have SNPs that affect transcription and SNPs that affect cancer. And of course, this is asking quite a lot. So we go from 450 genes to 137 eCSGs in the genome. So we're really concentrating on a very specific class of genes. So in the P53 pathways annotated by KEGG, there are 52 P53 pathway genes that have at least one eQTL. Of those 52 genes, 5 of them have also been associated with cancer GWAS. So we have 5 genes out of the 52. Now, this represents an 8-fold enrichment of eCSGs compared to all the ones found in the 11,000 genes that have eQTLs. So again, we're in 7 to 8-fold enrichment. And if we do the pathway analysis, only P53 is enriched in these types of eCSGs and no other pathway. And again, it associates with a broad spectrum of cancers. So we've been using pathway annotation to define the important genes, but we all know who are in this field, there are certain core genes that are shown to be actual drivers, like Arnie had introduced before. So ones that we know when we mutate them, either somatically or in inherited predisposition syndromes, those are the genes that really truly cause cancer. So what if we now look genome-wide at those 500 or so that most people would agree have good evidence that they're cancer driver genes? And so the P53 pathway is pretty riddled with them. So there's 17 P53 pathway cancer genes. I think most people would recognize them, at least as annotated by KEGG. And we use the Sanger Cancer Gene Consensus Database to do this, but you can use Bert Vogelstein's as well. It's the same effect. So of the 17 genes, again, what dramatically 50% of those are CSGs, so have been identified in cancer GWAS. And so compared to the rest of the cancer driver genes, so 505, this is, again, a 7-fold enrichment. So even when we're focusing on those driver genes, we see the same enrichment. And again, P53 pathway, compared to all 214, is the most enriched. And again, it associates with a broad spectrum of cancer, and indeed is the broadest of all pathways. So putting this all together, if we just use KEGG, we have 10 genes in the KEGG pathway of P53 that associates with cancer, or that have been identified in cancer GWAS. What's really remarkable is that 9 of these have already been identified as driver genes using somatic genetic analysis. So those genes that are mutated randomly in your cell and then selected for in the tumor are the same genes that seem to be in inherited genetics, the ones that are malleable, the one that if they get a SNP, it will actually lead to differential cancer risk, which is something that we weren't expecting. And any way you do the p-value, this is very, very significant. So in conclusion, I've shown you that cancer susceptibility loci are enriched in P53 pathway genes. It's the highest and most significant enrichment compared to all 214 pathways. And indeed, we can do that using multiple pathway data annotation databases. And it associates with a significantly broad spectrum of cancer, just as we would have predicted based on the somatic genetics. Moreover, if we focus on those genes that also associate with altered expression of their proximal genes, we see the same type of enrichment, with P53 being the highest and most significant enrichment and associating with a broad spectrum of cancers. And indeed, when we restrict our analysis genome-wide to those genes that are known to be drivers, again, P53 is the highest and most significant and associated with a broader spectrum of cancers. Now, what's very surprising is that they're the same genes. That roughly all the ones that we're identifying in cancer GWASs, we've identified in whole genome sequencing of cancer tumors. But also, I think what was a little surprising was that we've seen no such enrichment in any other the diseases that have been tested thus far in the community, even though other diseases, other pathways are definitely affecting those syndromes. So what does this all mean? Again, if we go back to the BRCA example, these SNPs do not have the same effect as a BRCA mutation. Their penetrance, as we say, the effect and overall risk is lower. But the fact that we see such an enrichment, such a predicted enrichment of the P53 pathway means that we could start putting these together based on all this wonderful information that you've seen in mouse models and somatic genetics to actually inform risk assessment. So we can identify, there's a big debate in the field, do we do mammograms or not? Countries are stopping them. But potentially we can use this type of pathway information to inform who might benefit from that. Moreover, it seems that there are genes within the pathway that are highly sensitive to genetic manipulation, either those that occur randomly somatically or those that we're passing on to our children. And so, of course, this means that these genetically malleable genes might be also pharmaceutically malleable. So they might be the ones, if we want to affect P53 signaling, are the ones that we should be targeting. Moreover, in terms of biomarker integration into personalization strategies, we need to start incorporating the inherited genetics because, indeed, these are as you're going to hear from Chris, in the MDM4 gene, these are the ones that we're targeting. So, of course, finding those patient populations that are going to have the most benefit and the less side effects, incorporating this information could help it make this more realistic. And moreover, as we start manipulating the P53 pathway in the clinic, we're going to have to start worrying about side effects because, of course, a lot of cells utilize the P53 stress response, and indeed that has hindered a lot of phase 2 and phase 3 studies because they are so toxic. But if we start looking at the genetics in the broader population, and based on our observation that the P53 pathway primarily affects susceptibility to cancer and not other major disease groupings we've tested, including cardiac disease, this might help inform and predict and monitor potential side effects. Okay, so with that, I'll stop. And I want to thank Giovanni, who is the bioinformatician who did the analysis of the last section that I talked about, and Jorge Zeron, who really led the polymorphic response element study, and of course our collaborators, who I want to make sure, Douglas Bell and Xuting Wang at the NIH, who's been, all of this work has been in close collaboration with them. So with that, I'll stop and take questions. Thank you.